Welcome to this webinar on particularly cell sample handling. I know the title was sample, something with sample, but it's especially cell samples. I'm really happy to have and introduce Jonathan Dodd here with us today. Jonathan is Senior Manager of Assay Development at Revvity and a trained T-cell immunologist, and he has spent a number of years perfecting assay design, and he has had a particular focus on tuberculosis diagnostics. So any questions you have on cell samples after this, please post them in the Q&A section. From Mabtech side, we have Tyler Sandberg. He is our product manager for Assays, and he will begin this webinar. Please press continue to share, Tyler. He will begin this webinar by giving you a short overview on cell-based assays, and then talking about our in-house Mabtech's recommendations for and tips and tricks on cell handling. Then the main part of the webinar will be Jonathan's. He will tell us an intriguing story on storing blood, on aged blood, and what happens with the different components. My name is Gertow. I'll be your host for this webinar. We aim for a 30-minute presentation, leaving 15 minutes for your questions. Some of you have already posted questions when you signed up, but please also add questions in this Q&A function you have in the bottom of the Zoom screen. I will keep track of them during the webinar, and then I will ask them to Tyler and Jonathan during the last 15 minutes. And we will try to answer as many as possible. A final piece of information, we are recording this webinar, so you know that. And we will also post it on our YouTube channel. And you will get a link to it afterwards if you can't stay the whole time, for example. That was it from me for now. I have had my fifth cup of coffee for today, so I'm ready to get into it. So let's begin, Tyler.
Nice. Thank you for the introduction, Jens. And yeah, we're excited about this topic today, about good cell quality, giving you good data. And we'll focus on a few different methods today, just so I'll give a short little introduction. The first one that we'll talk a bit about is FluoroSpot. And so this is the multiplexed version of ELISpot. So rather than looking at a single analyte being secreted by your cells of interest after being stimulated with your antigen, your peptide pools, here you can multiplex up to 4 different analytes in the same well because we're using fluorophore detection. So that allows you to look at the number of responding cells as well as the co-secretory patterns. So you can look at these monofunctional cells as well as these different polyfunctional cells. And in our systems, you also get the added benefit of being able to see the relative amount of analyte secreted. So in a typical FluoroSpot, this is what a typical FluoroSpot readout can look like. So you have these 4 different fluorescent-based images that are then overlaid, allowing you to look at these polyfunctional cells and look at these different subpopulations. So based off of the secretory profile, you can understand if you had more of a Th1, Th2 response, or if you're throwing in granzyme B or perforin in there, then you can even look at the cytotoxic T cells. These assays are great for cytokines, but also equally applicable for B-cell, ELISpot, and FluoroSpot. The other assays that we'll focus on today are these different flow-based assays. And these are quite powerful to use in parallel with FluoroSpot to give a full picture of the T-cell response. So the first one we'll talk about is intracellular cytokine staining. So this is looking at the cytokine production in these cells following antigen-specific stimulation, very similar to FluoroSpot. But here, you're not allowing the cells to actually secrete those analytes, those cytokines that are being produced in response to the antigen-specific stimulation. So you're actually blocking this with a Golgi stop, for example, but that also limits the amount of time that you can incubate your cells with the stimuli, for example. But then you're staining for these different cytokines intracellularly and analyzing on a flow cytometer. The other flow-based method, which has become quite popular in the last few years, is the AIM assay, activation-induced marker assay. So rather than looking at cytokine secretion or production, you're actually looking at the upregulation of different activation markers on these cells. So common ones are CD69, CD137, and OX40. after the antigen stimulation. So this has the added benefit that you can increase the time of stimulation similar to ELISpot and FluoroSpot. But used together, like I said, you can get a very full picture of your T cell response. And regardless of which assay you're using, there are some key variables to consider in all of these functional-based assays. And we won't talk a lot about it, but I wanted to make sure it was listed here because it is an important factor to consider is the choice of your media and serum supplementation, whether you're using serum-free medias or using FBS supplemented RPMI, for example. And regardless of which one of these that you're using, you want to make sure that you've pre-screened these before use in, for example, a clinical trial or your larger study. Just to make sure that these serum-free medias or the FBS batches that you're looking at aren't inducing unspecific responses from your cells. So that's sort of all we'll touch on about those things. You could have a whole nother webinar talking about different serum-free medias and FBS and things like that. But today we want to really focus on cell quality. Jonathan and I have talked about previously when we've been planning this webinar that the same concept of garbage in, garbage out applies to cell quality and your data quality. If you have garbage quality cells going into your assay, the data that you're getting out of it is going to be quite questionable and maybe not the most reliable. So there's definitely a lot of considerations to have when you're planning your assays and to get the best cell quality, and you generally want to have a viability over 90%. So this can be done by optimizing your cell handling and counting, the time between blood draw as well as PBMC isolation, the actual isolation protocol, the freezing and thawing SOPs, as well as including resting into your thawing process before analyzing with FluoroSpot or flow cytometry. And this image on the right just shows how important it is to have a good viability with your cells and good quality cells. Because as you see, as we have a decreasing viability of cells by adding water into these wells, you're getting a much fewer spot-forming units from PHA stimulation. So you really do need a good viability for these assays. So the first things to consider are how you're prepping your PBMCs for use in your experiments. Probably the most common ones that are used are the separation tube techniques. So doing like a fecal overlay where you're diluting your blood 1-to-1 in PBS and carefully laying that over a fecal or lymphoprep and centrifuging to be able to separate red blood cells and other blood components from your PBMC fraction that you can then use in downstream analysis, either fresh or you can cryopreserve them for later analysis. This is what we're mainly using in-house at Mabtech. So you do have to be quite skilled to do this. You don't want to just let anyone in the lab going and doing this right at the beginning of a clinical trial. You want to make sure that people are well practiced and rehearsed in doing these. Another really great alternative is the use of CPT tubes. These are a little bit pricier, but they require no lab experience and can be done on site. So if you have a multi-center study where blood is being collected at all these different places, these are really a great option to be able to spin down the blood directly after the blood draw to be able to get that PBMC fraction out of there while separating the rest of the blood components out of there. And then these can be shipped to the analysis center either overnight, or you can check the protocols in the data sheets for those products. But regardless of which method that you're using, you might need to tweak things here and there to get that 90% viability. We show exactly how we do things on our website. We have a nice YouTube video showing how we do PBMC isolation, as well as a step-by-step protocol online. The next thing to consider is the time from blood draw to PBMC isolation. And it's pretty well accepted in the field that 8 hours is that sort of maximum time that you want your blood just to be sitting before isolating your PBMCs. And this was a study from 2007, where regardless of the method of PBMC isolation or the different anticoagulants that were used, the biggest factor here in spot forming units and the functional response of your T cells was the time was leaving your cells more than 8 hours. So here they compared 8 hours and 24 hours, and you see a drastic decrease in the spot forming units at the 24-hour time mark compared to isolating those before 8 hours. And probably the biggest enemy that becomes activated or sort of has a negative effect on your T cells after 8 hours is the granulocyte fraction. And these granulocytes are difficult to remove from your PBMC fraction after 8 hours because they change their buoyancy. So they're harder to eliminate through gradient centrifugation after that. And this was a nice study from 2005 where they were showing that increasing fractions of granulocytes actually has a negative effect on the spot quality that are being used, that you're using to generate your data, as well as the spot numbers. So increasing granulate contamination actually led to a lower number of spot forming units. So showing that they've already done their work, even if they were potentially removed later. So now that you know that you want to hopefully isolate your cells within 8 hours, the next things to consider are how you're cryopreserving as well as thawing your cells. So there's a lot of different ways and different freezing medias that you can use. During my PhD, we were using full FBS with 10% DSMO or DMSO. At Mabtech, we use RPMI with 20% FBS and 10% DMSO. But there are also plenty of commercial freezing medias available. Same sort of thing goes here. You want to make sure you're screening these methods and making sure you're not getting any of these that are creating unstimulated responses in your cells. When it comes time to thawing, a big thing is to not shake your cryovials. And I was very guilty of this during my PhD because no one told me this. So when you're thawing your cells in the vial, you actually have all these small ice crystals. And if you're shaking it around, you actually risk puncturing the cells and leading to worse recovery. And this can lead to more DNA being released into the samples and creating all of these clumps. So try not to shake. But another way to circumvent excessive clumping that you might see in your samples is adding DNase or benzonase in your thawing media. Finally, a final point to consider is adding resting cells overnight after thawing your cryopreserved cells. So this can be done in a high density concentration of cells, so 1 to 2 million cells per mil in your same supplemented media with the tube unscrewed a little bit so you can get good gas exchange between the cells in the incubator. And those can be left overnight. And there was quite an elegant study published in 2014 that really showed the benefits of adding this overnight resting, or what we call resurrecting your cells after cryopreservation. So in this study, they used cryopreserved cells and they tested them in ELISpot 0 hours after thawing, 18 hours after thawing, or 22 hours after thawing. And at the 0 hour time mark, so directly after thawing and putting them onto the ELISpot plate, you're able to detect a certain number of antigen-specific T cells, but it's statistically increased in magnitude if you waited 18 hours or 22 hours after thawing. So keeping them in the incubator overnight is allowing all of those maybe stressed cells after thawing to let them die and sort of let them go away so that the cell fraction that you're remaining with after that resting period has a really high quality, high functioning T cells that can be used in your ELISpot or FluoroSpot assays. There's also the added benefit of adding this resting step by getting rid of any sort of background artifacts. So this is from the supplementary material of that paper, where they showed that adding that 18 or 22 hour resting period can also help really clean up the background. So when you're only adding PBMCs and media control wells, you're getting a much better background, so you can really get reliable counts from your antigen-specific cells. And those are sort of tips and things that we've learned over the years from different ELISpot research group workshops that we've had, different in-house experience that we have. And now I want to hand it over to Jonathan to talk about a study about using aged blood and some different ways to assess those. So I'll let...
Thank you, Tyler. I'd like to start by thanking Mabtech for inviting me to speak. My name is Jonathan Dodd. I work for a company called Revvity, which is a multinational company. I work in a UK site in Oxford, and I help operate an on-demand CRO, which has worked in many clinical studies, mainly on the ELISpot, FluoroSpot, and flow cytometry platforms. Typically in vaccine trials. And one of the major findings that we've made over the years is that one of the major factors in determining the quality of your data in those studies and those trials is the quality of the cell sample. So I will just, there we go. Oh, okay. When we often talk about blood and the impact on PBMC quality, we often talk about neutrophil degranulation. But blood is much more complex than that. At last count, there were 238 molecules associated with whole blood samples and 27 cell types. So there's a large heterogeneous mix of humoral and cellular factors in close, tight homeostatic regulation. And as soon as the blood leaves the circulation into a tube, it begins to degrade, and those homeostatic mechanisms are lost. And so it's known that as soon as the blood is collected, it begins to degrade. There are various critical components that change throughout this process, some of which are listed here. So we have critical electrolytes such as sodium and potassium, which dictate the water potential between the cells and the plasma. We also have respiratory gases such as oxygen and carbon dioxide, which are critical for metabolism. Together with pH buffering effects of the albumin and globulins, the cells use up the fuel that remains in the plasma. So levels of glucose decrease very rapidly in collected blood, and waste products increase. On top of this, you have critical regulatory molecules such as hormones and signaling cytokines and growth factors that are required to maintain the cellular health of the components within the bloodstream. And so it's logical that as soon as the blood is collected, those processes begin to break down. And the longer that the blood is left before it's processed, the more degraded and changed from the in situ situation it becomes. So looking at a kinetic analysis of the degradation profile of blood, we know that there's this 8-hour safety window in general in order to, before you have to process, but different factors within the whole blood sample degrade at different rates. So for example, it's known that in order to be able to separate the serum and plasma from the remainder of the blood sample, that should occur within 2 hours in order to preserve analyte integrity. Whilst the cellular factors can degrade within 24 hours, molecular factors such as the DNA and RNA are typically a little bit more stable. So an RNA profile and transcriptomic profile is generally lost within about 24 hours, but the DNA profile can last up to 360 hours, making it much more reliable for molecular analysis. And likewise, methylation is DNA methylation is more stable than the RNA equivalent. Plasma proteins and critical metabolites such as glucose, these typically degrade within 24 hours, maybe up to 48, depending upon the specific molecule. But essentially, because of these significant changes in the biochemistry of the blood sample in the first 24 hours, the cellular components begin to show significant signs of change within that same time period. And a few different studies have been reviewed and published on the temporal degradation of the hematological and biological parameters in blood. And one of the studies is shown here. And what this looked at is a variety of cellular components on the left and humoral components on the right. And they demonstrated that within as few as 4 hours, you're starting to see significant changes in both. So in particular, the total lymphocyte count, which is this green line here, and the absolute neutrophil count both begin to change within a few hours of collection. Likewise, critical metabolic factors such as glucose begin to deplete very, very quickly as the cell use up the remaining fuel that's present in the sample. And ions such as sodium and potassium begin to change, such that by 24 hours, glucose levels have decreased very significantly by about 25%, indicating significant metabolic activity and degradation. Sodium levels have decreased by up to 15%. Potassium levels changed dramatically by up to 50%, indicating cellular lysis and leakage. And corresponding to this, by 24 hours, you're starting to see significant reductions in the lymphocyte repertoire and the lymphocyte counts that are present, affecting both your T and B cell repertoires. And the absolute neutrophil count has dropped significantly also. So it's clear that by 24 hours, the cellular and humoral components of blood are significantly altered after collection. Likewise, other studies have looked at the actual functional profile of the cells that remain in the blood after delayed processing. And what has been found is that although the PBMC subset proportions and their viability remains largely untouched, you can expect an approximate 20% reduction in viability within the first 72 hours. The functional profile of the cells changes very quickly. So, for example, chemokine receptor expression on the PBMC decreases very quickly, which suggests that they're responding to the remaining chemokines that are present as humoral factors in the whole blood sample. Likewise, neutrophil contamination within the PBMC layer is caused due to changes in their density because they undergo apoptosis, activation steps as they respond to the changing environment, which obviously, as we know, interferes with downstream assays. Moreover, activation markers on all of these cell subsets begin to decrease, again suggesting changes in the transcriptomic and in the functional profiles of the cells that remain viable. So overall, we know that although the viability of the cells remains largely intact, the functional profile of the cells changes dramatically and rapidly. So within Revvity, our on-demand CRO called Certimmune has used PBMC stabilization reagents, and one here called T-cell extend. And these agents are useful because they enable the cleaning up of the sample to improve the quality of the PBMC layer prior to use in your downstream assays. And T-cell extend is a reagent that's added at the point of blood processing, so it's not required to be used at the site of collection. And it's added, and it acts by cross-linking the granulocytes and red blood cells that can contaminate the PBMC layer upon density centrifugation. And by cross-linking them, it increases their density, meaning that they are removed upon centrifugation. So this reagent has been validated in our ELISpot platforms for up to 32 hours. And one of its major advantages is that it doesn't interact with the adaptive immune cells, the T cells and B cells that are present in the sample, maintaining their integrity and functionality for measurement in downstream application. And as I said, it's used at the processing lab, so there's no requirement for training or extra steps at the enrollment sites. So the study that the data from the study that I'm going to show you took place about a year ago, and we were interested in performing a sort of a deeper dive into how PBMC stabilization reagents can protect the quality of the PBMC sample in blood that's had its processing delayed. So one of the obvious questions that we had was how well does it protect the viability and functionality of blood that's had its processing delayed? And more interestingly, does its protective effect occur across all different assay platforms, so ELISpot, FluoroSpot, flow cytometry, equally, or do we see protection on one platform but not on another? And further, do we see differences in protection across different cell types? So do CD4 T cells, are they protected more than CD8 T cells, or Th1 responses more than Th2? So in order to address these questions, we performed the following study protocol. We took blood from 52 healthy volunteers and we paired them up, each pair having a fresh sample that was processed at 0 hours. And then it was paired with blood from the same donor that was delayed in its processing by either 24, 32, or 72 hours. For each pair, one tube was not treated with the T-cell extend reagent, or it was using our in-house validated protocols. The blood was then processed by density centrifugation to extract the PBMC layer. These were then prepared for culture on two platforms. We used FluoroSpot plates supplied by Mabtech, and we used flow cytometry. So we cultured the cells prior to their analysis by antibody staining. We used 4 experimental conditions in the assays, 2 controls, media as a negative control and PHA as a positive control, and 2 model antigens that we use in-house regularly, purified protein derivative and spike protein from the Wuhan strain. So for both platforms, 250,000 cells per well were used. Each condition was performed in triplicates, and the incubation time was 4 days. And then after this 4-day period, they were read using manufacturer's protocols or in our own in-house protocols for the flow cytometry. So we measured the viability of the cells prior to their use in culture, and we found very little difference between the different conditions. We only saw approximately a 20% reduction in viability between 0 and 72 hours. However, we also measured the viability at the end of the culture period in both platforms, and it was here where we started to see significant differences in the performance of the cells. So in the graph on the left, we're looking at the kinetic of PBMC viability at the end of the culture period. On the x-axis, you have the sample time before it was processed, and on the y-axis, you have the viability of the cells. And the graph is split into 2. The left-hand side, the cells were not treated with T-cell extend, and on the right, they were. And as you can see, the viability of freshly isolated cells at the end of the culture was approximately 60%, and this was maintained in samples that were where processing was delayed for 24 hours. We then saw an approximate 23% reduction in viability with cells used after 32 hours, and if the cells were left until 72 hours, the viability collapsed. When we used extend on those same samples, we found that the significant difference that occurred was that at the 32-hour time point, we saw a protection of PBMC viability by the T-cell extend. However, this was lost by 72 hours. We then wanted to know whether this protection that we observed at 32 hours was antigen dependent or whether it occurred across all experimental conditions. And so the graph on the right shows the breakdown of this of the cell viabilities across the 4 different antigens. And what you can see is that there was a statistically significant increase in the viability of the cells when using T-cell extend across the media, PPD, and spike conditions. And so it suggested that the protection that this T-cell extend was offering on the viability of the cells was independent of the antigens used. We then looked at the performance of the assays on the FluoroSpot platform. And because of time, I'm only going to show you a subset of the data. But what we found was that for PPD stimulation, we saw a reduction in interferon gamma spot counts in the 24-hour samples, which was then increased dramatically in the 32-hour samples. So by 32 hours, we were losing almost all of the interferon gamma response in the cells upon stimulation with PPD. And I should point out that this is antigen specific. The media control has been deducted from these values. When we looked at the protection offered by T-cell extend, we found that there was complete protection in the 24-hour samples, and there was partial protection of approximately 60% in the 32-hour time point samples. However, there was no protection offered by 72 hours where we saw an ablation in the interferon gamma spot count. If we compared this to the spike antigen responses, we saw an approximate 70% reduction at 24 hours in the absence of T-cell extend, and an ablation, as we observed previously, at 32 hours and 72 hours. And when we compared this in the presence of T-cell extend, we found partial protection of approximately 40% at 24 hours, but only 20% at 32 hours and no protection at 72 hours. So it suggested that there was protection on the FluoroSpot platform, that to a certain extent it was antigen specific. We then asked the question whether it was specific to interferon gamma. And so in these graphs here, I'm showing you the combined cytokine spot counts for IL-4 and IL-5 in these assays. And what we found was that in the absence of T-cell extend, upon spike stimulation, we saw an 80% reduction in spot counts at 24 hours, and they were lost by 32. In contrast, we saw a protection in the IL-4 and IL-5 spot counts in the presence of T-cell extend, which was small but noticeable at 32 hours, but lost at 72, suggesting that the protection wasn't simply restricted to interferon gamma, it also extended to Th2 cytokines also. Because the spot counts for the Th2 cytokines were a lot lower than interferon gamma, we also checked the positive control condition, and we found partial protection in that scenario also, in that we saw a 40% reduction in 24 hours in the absence of extend, and this again was lost by 72. And in the presence of T-cell extend, we saw a partial protection of approximately 20% at 24 hours, about 10% at 32 hours, and 0 at 72 hours. So it did suggest that PBMC stabilization reagents could protect the cytokine responses on the FluoroSpot platform for both Th1 and Th2 cytokines. We then compared the data on the flow cytometry platform. So we use 3 separate metrics for T-cell activation on the flow cytometry platform. We use proliferation as measured by CFSE dye dilution. We use activation markers, exhaustion markers, and we also use cytokine production. What I'm showing you here is the percentage of cells that are proliferating and expressing CD25, because we consider that the gold standard. The left-hand graph is showing you the responses of CD4 T cells, and the right-hand graph is showing the response of CD8. And as you can see, in contrast to the FluoroSpot platform, when we measured responses on the flow cytometer, we got near ablation of the response observed in the fresh cells by 24 hours, and this was not rescued at any other time point. In the presence of T-cell extend, we saw no protection whatsoever. We could not rescue the percentage stimulation in any of the time point samples measured used in this study. This was not restricted to the CD4 T cells. When we measured the same analytes on CD8 T cells, again, we saw an ablation of the response by 24 hours, which was maintained at 32 and 72 hours. And again, in the presence of extend, we saw no protection whatsoever on the flow cytometry platform. So it was clear that although there was protection offered on the FluoroSpot platform, it was not extending to the flow cytometric platform. And just to show you some examples of the kind of data that we were seeing, these plots on the left are showing the different combinations of the activation parameters that we use. So you have proliferation as measured by CFSE dye dilution on the x-axis, CD25 expression on the y. In the middle column, you've got activation and exhaustion markers. And in the third column, you've got activation markers and interferon gamma secretion on the x-axis. Each row is a different set of conditions. So you can see these are the top row is freshly isolated cells that have not been treated with T-cell extend, and we can observe strong proliferation, strong activation and exhaustion marker expression, and good interferon gamma expression. However, when we use the sample that had been processed after 24 hours of storage in the absence of extend, we observed none of these features. They were all largely ablated on the dot plots. And surprisingly, when we added T-cell extend to try and protect these cells after 24 hours, we saw no rescue in the profile that we observed in the fresh cells. Again, confirming that 24-hour-old blood loses almost all of its functional biomarker expression on the flow cytometric platform. So we've spent quite a long time trying to understand why we see this difference on the FluoroSpot and the flow cytometry platforms, and we believe that it's due to a differential sensitivity in the cell viability and integrity requirements that the two platforms have. PBMC stabilization reagents may be able to preserve the viable cytokine secreting cells for the FluoroSpot platform, but they're unable to maintain the membrane integrity, the surface marker stability, and the transcriptional translational machinery that flow cytometry requires in order to generate a meaningful result. Flow cytometry often uses surface marker expression, and these appear to be shared or proteolytically cleaved and internalized on what we assume to be a rapidly decaying cell membrane. Moreover, the method of cytokine capture differs between the two platforms. So whereas FluoroSpot captures an accumulating analyte signal, which will increase even if the cells are partially compromised, on the flow cytometry platform, which relies on permeabilization and intact organelles in order to function, these are likely of degraded, leaking the analyte and reducing the signal. The metabolic decline that we observe in the blood sample is likely leading to a certain level of T-cell exhaustion and antigen unresponsiveness, which is leading to reduced proliferation, reducing the effectiveness of the CFSE dye dilution reagent and also the activation markers. And likewise, the reagents that are used in flow cytometry, such as the Brufeld-9 monensin, may stop working properly because they are no longer able to interact with the organelles, the Golgi and the ER, as they can on fresh cells because the cells are rapidly decaying. So we believe that the differential sensitivity that we've observed here results in the FluoroSpot platform being more tolerant to lower cell viabilities and only partial cell activation and cell integrity, whereas flow cytometry, which requires a higher quality, more intact cell for accurate quantification, is much more susceptible to delays in blood processing. And I'd like to stop there and address any questions that you may have.
Thank you, Jonathan, and thank you, Tyler. The floor is open for questions, and we have several questions already posted, as said. But let's begin with the most pressing ones that have come through the Q&A and chat so far. And one is, so you showed, Jonathan, stability data for blood. Do you have, or can you guess if these features are similar for buffy coat?
We've never formally empirically tested that, but I would assume so because the overall decay process, the storage process would be similar. The mechanisms that we believe are important in the loss of cell functionality should be intact with a buffy coat. So I would say yes, but we've not empirically demonstrated that.
Right. Some questions that came in previously were about resting, and you addressed resting, Tyler, in your session, but it was about, like, I think you talked mainly about overnight resting. Can one rest cells for a shorter time? Have we tested 4 hours, 30 minutes? Do you have some input there, Tyler?
After thawing, generally, we've done a mixture of like either using overnight resting, or sometimes we'll just go straight into the assay if we're pressed on time sometimes. But for some of these more important studies, it's definitely good to assess the outcome and assess using the antigens of interest that you're going to be using. So you can see this, but you're definitely going to see an improvement, especially with that background background noise that you're seeing if you're not using any of that resting, because that, freezing and thawing cells can be quite stressful for the cells. And if there's maybe something that's been going wrong, there could be cells that have already committed to dying that's going to affect your later ELISpot or FluoroSpot assay. So generally, it's recommended to keep that overnight resting there after cryopreservation.
Right, and an associated question to that is that for room temperature or 37 degrees? It's
at
for
37
degrees. And another associated question is how to prevent PBMC clumping. And you mentioned benzonase and DNase. Can you elaborate on that?
Yeah, so that's a step that you can add to your thawing procedure. So it doesn't need to be present in the media for the entire protocol, but just in that first step when you're adding your cells dropwise to the warmed media, you can add benzonase or DNase there. And then do that first washing, and then it should have removed all of those clumps that are present.
I would say that overnight resting is particularly crucial if you have low cell viability. So anything below 80%, you should definitely be resting them because you're then selecting on the viable cells after your overnight incubation.
Right. I remember during my PhD, we were concerned about doing overnight resting because we usually saw a decrease in number of cells before and after that overnight resting. But those cells that were present after that overnight resting were much more functional and much better for answering our specific questions. So it's definitely the beneficial one.
A question that came in through the Q&A function is, do you see similar results if the PBMC are frozen? I'm not sure if this is directed to Jonathan or Tyler.
So the use of the PBMC stabilization reagent is only with whole blood. So if you've already processed your sample and cryopreserved the PBMC, there's no benefit to using the stabilization reagent at that point. The only caveat to that is that if you have a particular poor quality sample, if it is heavily contaminated with blood, then that may warrant a repeat process with the PBMC stabilization reagent. But that is the only time when you would even consider doing that.
And an associated question also to maybe the Buffy coat and other types of samples. I mean, we have had questions coming in regarding ELISA and lateral flow and other like plasma and serum-based assays. Do you think, if you don't have empirical evidence, that aged blood have an impact on those types of samples as well?
Yeah, they do. So obviously most people, particularly in the vaccine space, they're very interested in cells, they're interested in adaptive immune cells or serology. So each component of blood has an individual stability profile, and so they decay at different rates. So antibody responses are generally a bit more robust than cellular responses for obvious reasons. There's a lot more that can go wrong in a cell. But nevertheless, just as a good practice, you should be processing your sample to cryopreservation or use as soon as possible after collection. The longer you leave it, the less in situ it becomes. And so the less reliable and less accurate your data becomes. So I think irrespective of the component that you're even looking at, you should definitely be using it or processing it as quickly as possible.
Right. Then a question on analysis. Let's say you have run your experiment and you actually had low variability from blood. So you have really small responses through ELISpot and FluoroSpot. Do you have any idea or tip on a method of normalization to ensure appropriate quantification of your test wells?
Yeah, so if you've got low viability, before you run your assay, then what I would suggest is resting it. As Tyler said, you will maximize the functionality of the remaining cells. It will also reduce the non-specific background. To normalize it, what I would do if you're using ELISpot or FluoroSpot platform, you normalize to a number of cells. So SFU per million is typical. In flow cytometry, what you can do is a similar thing. It depends on what you're looking at, but you can, obviously, if you're measuring a functional response, you can measure it you can normalize it to a number of cells. So a percentage per 1,000 or a percentage per million, whatever's more convenient. And that's the most appropriate way to normalize such that you can compare sample to sample that may have differing qualities. But I would also recommend, because we've had to do this in the past in various clinical studies, I would recommend quarantining any data where there is low viability samples being used, because you can never be 100% certain, even with the best practice, that your data is reliable. So I would always quarantine it and then analyze it quite carefully, and even put in a comments saying certain samples had low viability, and this is what we've observed, so that it's clear that the data may not be as accurate as other data points in your study.
Right, and a question connected to that is there a way to increase viability at the end of simulation? For example, pushing it from 60 to 80%. Handling tips, maybe.
After stimulation.
Yeah, that's a tricky. Tricky. I think you've got to set up your experimental conditions as optimally as possible. As already been mentioned, supplementation of the media as appropriate. You would have to validate that for your particular assay of choice on your particular platform. But obviously, it relies on basic things like good pipetting, aseptic technique, those basic principles would maximize the quality of your prep.
Yeah.
And it would do so at the start, so it would do so at the end.
Great, thank you. So we are actually at time now. It's 5:45. So we want to respect your time. The webinar is over. Jonathan, Tyler, and I, we have another 5 minutes. If you want to stay, please do. We have a few more questions that we will try to answer in the last 5 minutes. But officially, webinar is over. Thank you everyone for joining. So just for the last couple of minutes, Jonathan and Tyler, if we could just answer also. Tyler, would you please repeat why granulocytes can cause problems after 8 hours?
Yeah, so after 8 hours, the issue is that you can't, it's harder to remove them from the, or when you're doing your PBMC isolation, when you're doing the FICAL, it's when you're doing the overlay, the granulocytes change their buoyancy. So they're actually not going to go through the FICAL layer after 8 hours. So then they're going to be contaminating your T cell or your PBMC fraction. And that you've seen in the slides that you can have degraded spot quality in the ELISpot results. And then as far as affecting sort of functionality of the T cells, maybe this is a better question for Jonathan, if you have any input there, because I'm actually not quite exactly sure why it's happening.
I think granulocytes are one of the main contributors to the degradation in the PBMC layer. Beyond the decrease in density and their contamination into the PBMC layer, they're just a very, very angry cell. It's like they're a cell that's had like 20 cups of coffee as opposed to Jens's 5. They're extremely sensitive to biochemical change and signaling. So they chuck out DNA, they secrete lots of inflammatory factors, they die very quickly, they degrade quickly, they just hyperactive, and so they can be the source of a lot of problems in a blood sample that's had delayed processing. So again, it's one of the reasons why you want to process it as quickly as possible, and use stabilization reagents where you can.
Yeah.
Let's dive into the most pressing questions that came up already in your presentation, Tyler, regarding resting. You mentioned resting overnight. Can one rest for a shorter time window? Is it after cryopreservation, or is it also like, should you rest after FICAL?
Yeah, so the study that I showed was looking at cells after cryopreservation. So that 0, 18, and 22 hours was after thawing. So these are cells that were processed, I believe, within 8 hours and cryopreserved. But I have to double check in the study. So yeah, but I mean, that study, they didn't look at any other time points besides that. And we've not done any extensive studies at Mabtech to sort of look at the effects of these different hours, incubation or resting strategies. But it would be quite interesting to almost get like a sort of kinetic-like study to be able to sort of look at every hour and sort of see what that sweet spot is. So it could be beneficial to assessing it yourselves before you're committing to a larger study if you're going to be using cryopreserved cells.
Can I comment on that?
Yeah.
So we've studied this, both the impact of cryopreservation and thawing and resting, but also we've used it in ex vivo culture assay validation. And what we've found is, I think the cryopreservation is quite well known, that if you rest the cells overnight, that you select for the viable cells in your sample. And we've seen the same thing. But also, if you ex vivo culture in order to generate T cell lines, for example, if you rest the cells prior to performing an ELISpot, for example, to test for reactivity, we saw an ablation of background if you rested, whereas if you didn't rest, we saw quite high background. So actually, I think resting can be a critical component of a good assay design.
An associated question to that, if you want to rest overnight, then how do you do it? How do you go about it?
Yeah, so I mean, you want a high concentration of cells. So I think in the slide I wrote 1 to 2 million cells per mil. and usually in a conical tube. So those cells are really nice and close together because you want that cell-to-cell interaction to make sure they're getting correct signaling and talking to each other sort of, and they'll feel the best way that way. They'll be happiest that way for the next day. So you usually do that in a conical tube, and then you have the lid screwed off just a little bit so you get gas exchange. You could also do this in sort of fax tubes because they also have sort of a looser fitting lid. But as long as you have that high density of cells, that's what we've seen to work well.
Thank you. So we have another question that was posted previously regarding lateral flow immunoassays. What we have talked about here today, can that be applied to lateral flow as well? What do you think?
We've never tested lateral flow assays. If it's a cytokine or a humoral factor that you're measuring, then I think you would have to validate the decay rate of the analyte that you're specifically measuring. So again, performing a similar study to the way we have here in just titrating the delayed processing time. Yeah, it will depend upon the inherent stability of the analyte.
Yeah. Another question on blood. Do you guys have any experience using CBDA blood for ELISpot? CPDA.
Yeah, you shouldn't use them. So I think they contain adenine. You shouldn't use those. They can interfere with the functionality. I mean, from T cells, I'm a T cell immunologist, so certainly for T cell responses, there is a known impact on the functionality of the cell. So I would avoid those and use lithium heparin as a first choice.
Right. We are using Buffy coats mainly in-house from the blood banks, and I have to double check with our scientists whether those are CPDA collected from the beginning. I'm actually not sure, but we will come back to that.
So it should be EDTA tubes for a molecular analysis. If you're interested in the RNA, DNA, or transcriptomics, we would recommend lithium heparin if it's the cellular component, the adaptive immune cells are interested in.
So I just wanted to say to everyone listening, we're actually at time, and I want to respect your time. So we should end the webinar here. We have, Tyler and Jonathan and I have another 5 minutes though. So if you want to stay, we will go through a few more questions for another 5 minutes, but as of this, the webinar is over, and you will get a link to the recording as well as questions and answers afterwards. So let's take a few more questions. This one's for you, Jonathan, I think. Why did you activate the cells during 4 days in the plate?
Yeah, we get that one a lot. So 24 hours or 18 to 24 hours is typical for an ELISpot or FluoroSpot. The problem with that incubation time is that you're only going to activate the high affinity TCR expressing cells. So again, T cell immunologist, I can't speak for B cells directly. But we've validated different time periods, and we've found a significant impact of the incubation time in particular on the sensitivity of the assay. So if you extend the incubation time, you allow all of the moderate and low affinity T cells to also respond. So you get a more complete repertoire analysis if you extend the time. It also improves sensitivity if you increase the number of cells per well. So obviously 250,000 is traditional, but we've actually validated up to 1 million. So we looked at the maths of an ELISpot or FluoroSpot well, and a monolayer is about 768,000 cells. So you can actually fit way more cells on the well floor than we actually do. So we advocate more the merrier. If you've got sufficient blood in order to generate more PBMC, use more PBMC per well in order to generate the most specific, the most sensitive response. And it doesn't impact background if you add more cells or increase the incubation time. So they need to be specific. There needs to be a specific antigen for them to respond. Although I will say for ELISpot, background is a less issue than compared to FluoroSpot. If you're doing FluoroSpot, you definitely need to use a blocking component. So we would advocate human serum. which is what we validated for as a blocking component for human cells.
Following up on sample types, are there any differences between Na heparin and lithium heparin? Any max level for heparin? Do you know, Jonathan?
Yeah, so I can't remember the mechanism actually, which is going to annoy me now. But yeah, you use lithium. Sodium has an impact on the biochemistry of the sample, or more so than lithium. Yeah, I would. That's why lithium heparin is more common than sodium heparin. So yeah, I would recommend lithium heparin rather than sodium.
Another question on the CPT tubes, Tyler, maybe this is for you, but also Jonathan. Would those be the best option if you have to delay preservation? Like, can you centrifuge on site and then transport overnight and cryopreserve the next day? Is that a possibility?
Yeah, so as long as the site collecting has the correct centrifuge as well as centrifuge buckets, because these are a little bit taller tubes than normal blood vials, that can be done on site, and then you can sort of invert them afterwards, so you get the PBMCs to mix with the plasma so that they're getting all of the goodies that they need that are part of the cellular components. And then those are usually shipped. They can be shipped, I think, up to 24 hours. It says in the data sheets for the tubes exactly how long you can transport them, but they should be transported at ambient temperature, and then they can be cryopreserved on site later. But those are a very good option. It does require a little bit more of an investment in just making sure that you have that centrifuge on site, but they are really good options.
Great. Let's do one final question. Then everyone who we have not answered the questions for, we will get back to you through email with a possible answer if we have one. So this is a question that actually came also already at sign up, but now I see it here in the chat as well, which is about medium and serum-free medium. AM5 medium versus R10 media. The person asking here is seeing differences, high level of background with AM5. Do we have any idea what's up with that?
I can make a comment, and I think we've talked a lot about this at different ELISpot resource group workshops, that with a lot of these serum-free media alternatives, a lot of them potentially are trying to compete with each other and adding more additives to sort of better supplement these medias, and that could potentially be causing stress for some of these cells. And what we've seen, there was a paper published a few years ago looking at a new validated FBS, and they compared FBS supplemented RPMI with these different serum-free medias. I think they looked at CTL medium and AIM5, and saw quite a high amount of apoptotic, or like induction of apoptotic cells. So the cells are getting stressed and causing them to die when you're assessing in ELISpot. So we don't have any proof to sort of go into if that is the actual reason why you're getting high background, but we've seen that sort of trend compared to good validated FBS. I don't know if you have any other comments or experience with that, Jonathan.
Yeah, we've used ex vivo media, AIM5 and RPMI as well. Yeah, you can, because it's serum free, there is an inherent risk of increased background because you're not blocking the well, which is why you're likely not seeing it with RPMI supplemented with 10% FBS. Yeah, I would simply recommend blocking. Either add a human, if it's human cells that you're using, then add human serum either to at the point of block the well before you add the cells, or add it directly to your media that you're using. Add human serum if you're using human cells, bovine serum if bovine cells, etc.
Thank you very much. We're even 7 minutes over time now, so it's time to say goodbye. As said, I have I see your names here. Anyone who hasn't gotten your questions answered, as well as some of the ones that were posted before, we will get back to you through email. Thank you very much for today, and see you next time.
Yeah, and big thank you to Jonathan for joining from Revvity. It was very fun to do a co-webinar with you all.
Very much so. Thank you.